Hi everyone, it's Mr. Sinti, and today I'm interested in discussing with you how viruses, and in particular bacteriophages, replicate in bacteria. Because one of the things about phage replication in bacteria is that, first thing I want to say is that this particular type of replication has led biologists to a lot of great understanding. It was like one of the basis of the background of Hershey and Chase, which led to the understanding that DNA and not protein was the nucleic acid. So while phages are probably one of the most understood of all viruses, and again, one of the reasons is that they're the host of the phage is not human, and so we like to study bacteriophages, but they're some of the more complex. And so this particular discussion that we're going to have today is that we want to understand the two basic alternative mechanisms of which phages can replicate in a bacteria, and that is through what is known as the lytic cycle. Let me see if I emphasize this. The lytic cycle is going to be a part of our discussion, and lysogenic cycle. So just briefly, the lytic cycle is going to result in the rupture of the bacteria, basically the pinata-like breaking open of the bacteria, and the bacteria are going to die, and the phage are going to replicate. The lysogenic cycle is more latent, it's more temperate, where the phage becomes incorporated into the bacteria genome, and it doesn't destroy the bacteria immediately. And so we're going to take a look at both of those in this conversation. And so I find this picture to be very interesting. To me, this is a picture that is just remarkable. Scanning electron micrograph showing a bacteria, and these are the phages right here in particular. The phage is attacking the outside of the bacteria. And basically this part right here of the phage is a protein. And so this part right here is a protein, and the protein is attached to the outside of the membrane. Now inside, you can't see this, but it's injecting its nucleic acid inside the bacteria, and that's how it's infecting the host. And so, again, bacteria can infect eukaryotic cells, and viruses can infect bacteria, and viruses can infect eukaryotic cells as well. Viruses are very small, and what they do is they can attach to the receptor protein of the host cell. And so this particular video is on the cycles of how viruses replicate. You're like, well, why is it called a cycle? Well, I'll get into that, and it's the point of the video. Whereas the viruses will replicate in one cell, and then they'll break up, and then that replication will repeat again, and thus a cycle. And so right here, you could see, again, electron micrograph, a lot of great electron micrographs of a phage. So this is all protein right here. All of this outer capsid is protein right there in the tail fibers. And inside is some kind of nucleic acid, possibly it's DNA. And so this outer part right here is mostly protein right there. Okay? So that's the basic structure. And so these are called, again, as I was mentioning, bacteriophages, from the Greek meaning to eat a bacteria, or just simply phages in general. So this is the replication cycle of that. And so again, drawing here, we're looking at the protein of the head, sometimes a polyhedral right here with the DNA inside, right there. Then you have this sort of body right here, this sort of rod-shaped structure of protein, and then the tail fibers where it attaches to the host bacteria. Sometimes the names of them are not very glamorous, but they're like these T even series, T2, T4, like that. Again, electron micrograph of a bacteriophage. Okay, so let's get into the lytic cycle. So the lytic cycle is a reproductive cycle in which the virus replicates. Now, you have to know that the virus cannot replicate on its own because it lacks those basic mechanisms. It doesn't have any organelles. It doesn't have ribosomes. So if it doesn't have ribosomes, it can't make its own protein, nor does it have polymerase enzymes to replicate its own nucleic acids. So what it does is it attaches to a bacteria and it replicates inside, and that's known as the lytic cycle. And the end of the lytic cycle culminates in the death of the bacteria. So the last part of this cycle is the bacterium breaks open, or lysis, which then releases the phage onto other bacteria, which then infects others, and so it cycles and cycles. And so it's called lytic because the cell is, if you will, causing the bacteria to lysis. And so these two right here is the basis of the word. Now, a phage that does this is considered to be virulent. It's a term that means that it produces by the lytic cycle. In other words, it's going to cause death to the cell. And so in a sort of step-by-step explanation, the phage attaches to the host receptor proteins. The red right in here is the single chromosome that's in a bacteria, for example, E. coli. Secondly, the protein stays on the outside, but the green nucleic acid enters the bacterium, so the phage injects its DNA. Now, the early part of this infection, or lytic cycle, is that the phage will start to replicate its own DNA. And again, the only way it would be able to do that is if it uses the host cell's nucleotides. That's brutal, isn't it? It's like somebody coming into your house and eating your food. So basically, it's replicating its own DNA. And this is sort of a basic diagram, but then what it also does is destroys the bacterial DNA, and then it uses the cell's ribosomes to translate its own amino acids by reading the viral DNA, or messenger RNA, I should say, into viral protein. So in other words, it's replicating heads and tails and bodies until the bacteria becomes sort of assembled inside. So it's phage assembly. And then ultimately, the phages will break out and the progeny, or children, will come out and infect other cells. And so it ends in lysis. So this is a virulent phage. Okay, and so basically a same conversation, different diagram. It attaches to a bacteria. One of the first things that it wants to do is destroy or degrade the host DNA. You might be like, well, what's the purpose of that? Well, it then gets the ability to slow down the protein production of the bacterium, and then the full emphasis is that on the viral. And so you synthesize virus genome and you synthesize viral proteins, and then inside they're assembled, inside the bacteria they're assembled, and then ultimately the cell lyses and releases the phages. And so this is an actual photograph of a bacterial lysis. So this is the lytic cycle, the end of the lytic cycle, where 200 or so viruses go out and infect other bacteria. So this picture is this right here. So this and this are the same thing. Pretty cool, I think. So a couple more details about this. Okay, so say T4 is attaching to E. coli. It attaches to specific receptor proteins on E. coli. Again, phages are host specific. And then what they do is, again, eject the nucleic acid into the host. The protein stays on the outside. The capsid remains, again, on the outside, sort of like a ghost right out here. But inside, the progeny are being replicated. In other words, the virus DNA is replicating, the proteins are being replicated, and then to facilitate the lysis of the bacteria, one of the genes on the virus is to code for an enzyme called lysozyme, which, if you're familiar with this, it's found in human tears as well. We produce this enzyme. It actually weakens and destroys the bacterial cell wall. So with damage to the wall, simply osmosis, the water movement into the cell, will cause the cell to lysis. and that's how the viruses get out. I find that kind of interesting. So this is another reminder to us all to make sure they're drinking plenty of fluids when we have an infection, because ultimately it's the water that's going to kill the bacteria. So a phage has the potential to wipe out a whole bacterial colony in just a few hours. As you can see, it's pretty brutal. But the bacteria are not totally defenseless against phages. They can battle against them. And so natural selection would favor a bacterial mutant that has odd receptor sites on its cell membrane. Because if it has odd receptor sites, then the phage won't be able to bind to it. So let me come back here. So if a mutation occurs to the receptor site, the phage just won't be able to bind, and so that's good for the bacteria. Just wanted to say, so that's kind of a defensive mechanism. One of the most important defense mechanisms that bacteria have against phages is that they can produce a whole group of enzymes, and these are hugely important in molecular biology and biotechnology. Again, no hyperbole alert necessary for this. I am telling you that these particular enzymes that a bacteria produces, they're called restriction nucleases or endonucleases. These restriction enzymes can recognize certain sequences of DNA and actually cut. So they're like scissor-like enzymes. This is not a very good drawing of a pair of scissors. But these particular enzymes, restriction enzymes, that bacteria produce are capable of cutting DNA. Okay, and you're like, well, wait a minute. And they're called restriction because they cut only at specific recognition sites. You're like, well, if a bacteria produces this kind of an enzyme that cuts up DNA, aren't you worried that it's going to cut up its own DNA? Yeah, well, it is a concern. But what the bacteria can do is it can take its own DNA. So this is self-DNA of a bacteria. And what it can do is occasionally it can mark the DNA with methyl groups. This is a functional group called methylation. And when these methyl groups are found on the bacterial DNA, the restriction enzymes do not cut. They do not cut cells' DNA that has methyl groups. And so therefore they would only cut the phage DNA. And so if you're following this, let me go back just for a picture over here. So when the bacteriophage injects its DNA into the host, it's like, see how it's it appears as if the bacteria is defenseless. Let's go up here. But in fact, these restriction enzymes are in here and they cut up the phage DNA, and so they don't get sick. As it turns out, there's all kinds of different restriction enzymes. They have different restriction sites. I will have to refer you to a different video on that because this is such an important concept. But each particular species of bacteria have their own restriction enzymes and strains as well. So those are pretty important. And again, just give me 1 second on terms of why they're important. Because as humans, we recognize the fact that if we have enzymes, and we can extract these from the bacteria, if we have enzymes that can cut DNA, we can then cut any DNA from any organism and use it to genetically engineer that DNA into a plasmid or any sort of genetically modified organism. So it's the basis of transformations and genetic engineering. Huge. Okay, so a virus comes in and it injects its DNA into the host. Okay, so here's the phage DNA, and then the restriction enzyme comes along like this and ends up cutting it up, and so therefore it defends itself against the phage. Pretty interesting. So let's talk about the lysogenic cycle. So the lysogenic cycle, if you recall at the top of the video I was describing, there's going to be two types of cycles. Lytic cycles, when the DNA goes in there, causes the bacteria to die. In this case, when the DNA is injected into the host from a phage, here's the bacterial host, here's the DNA. what can happen is that the DNA of the phage can actually become incorporated into the bacterial genome. The bacteria have usually one chromosome. And so if it enters into this, it doesn't immediately replicate itself, produce proteins, and destroy the cell. What would happen if it inserts itself into the chromosome? It's kind of like a prophage, if you will. And I'll label it as that. Let me go back to black. It's called a because like not quite yet, a prophage. And so this is interesting. So as the bacteria divides, it'll also replicate the prophage. So you get a bunch of bacteria with this phage in here, but it's not destroying the cell. So it's not immediately causing the cell to lysis. An example of a phage that does this is a very popular phage called phage lambda, the Greek symbol lambda, which is used as a wavelength. So it looks like a regular T4 or T2 bacterial phage that infects E. coli, but it does not have tail fibers. So lambda phage. So it looks sort of like that, but no tail fibers. So that's lambda phage. And so this is capable of not immediately destroying the bacteria. So it's not therefore considered to be virulent because it's not killing the cell instantly. It's more of a temperate, so like moderate. So phages like lambda phage can use the lysogenic cycle, but I'll tell you, it ultimately is going to result in the lytic cycle, as you can see here. So in terms of steps, the viral DNA is incorporated into the bacterial DNA, and as known, as I mentioned before, as a prophage. And so it's a part of a genetic recombination into a particular site in the host chromosome. And then at this point, what it does is it doesn't replicate. In other words, it doesn't start producing, I shouldn't say replicate, it doesn't start transcribing into proteins that would produce phages. It basically stays in there, replicates along with the host. Okay, and so it can divide along with the bacteria, and therefore the DNA of the virus is passed on to the daughter cells. And so occasionally, though, and this is where it marks the doom of the bacteria, occasionally the viral genome, prophage, it exits from the bacterial chromosome, and then that's going to initiate the lytic cycle, and it's bye-bye for the bacteria. Okay, so let's take a look at this. So this switch from lysogenic to lytic, we don't exactly know all the circumstances that causes the prophage to then excise or be removed and then enter into the lytic cycle. It can be induced by environmental factors that cause the bacteria to be in jeopardy, and therefore the phage starts to break out. But in the lab, we could cause this by just shining certain types of ultraviolet light onto the bacteria, and that causes the phage to become removed. And so let's look at this in terms of sequential. It's a good diagram. So lambda phage attaches to a bacteria, it injects its phage DNA, and then watch this, it then incorporates into the host DNA as a prophage. Now again, this could then divide and divide and divide for a long period of time. It could stay like this. So temperate, doesn't hurt it, but then, some kind of ultraviolet light or environmental trauma causes the phage to be excised from the chromosome, and therefore it's loose. And so if the viral DNA is loose in the cytoplasm, it's probably going to get replicated by polymerase. It's probably then going to be transcribed into viral RNA, and the viral RNA is going to go to the ribosome, and it's going to use the host cell's amino acids, brutal, to make more viral proteins. And then, you know what's going to happen. This is going to assemble, lysozyme's going to break the cell wall, and then, ouch, the bacteria dies or lysises. So it's a combination of lysogenic mode and then into the lytic cycle. And so the lambda phage infects E. coli sort of demonstrates the whole temperate phage lysogenic cycle. As you can see, I'll start off in the center. So it's either going to be like the phage attacks the bacteria and it replicates and then the cell dies, or it could be that it incorporates into the prophage, again divides for quite a while, reproducing normally, copying the prophage into future cells, and then it'll break loose, and if it breaks loose, it might then go into the lytic cycle and destroy the cell. So I hope that was informative. This was a brief look at how phages reproduce in bacteria using either the lytic or lysogenic cycles. Thanks for watching. Well, you know something? Sorry about that. I forgot that. Sorry about that. I got your hopes up that the video was probably over, but in fact, it's not. And sorry about that. I want to show you just in an encore a little animation of what I've been talking about, just in case it wasn't as clear as it could have been. Sorry.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat. The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
And thus it's a cycle because it continues to replicate on and on again. And then lysogenic cycle.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
Okay, so that again, you're like, well, I think it's important sometimes to see a cool animation like that. It's really well done. So again, I hope this was informative. A discussion on the reproductive cycles of phages. Thanks for watching.